Good morning, all. We are moving towards the second day of Talking Science. So we have two sessions today. Now, Dr. P.A. Jose will be speaking to us, and in the afternoon, Dr. Shweta will be speaking to us. The speaker for today is Dr. P.A. Jose, who is a principal scientist at KFRI. Dr. Jose hails from Thrissur. He joined his first job at the Jawaharlal Nehru Tropical Botanic Garden and Research Institute, Palur, Thiruvananthapuram, in 1987. He obtained his PhD from the Kerala University in botany. He had attended an internship at the prestigious Royal Botanical Gardens, Kew, England, during 1993. He is now specialized on species recovery research of endemic and threatened trees of Western Ghats, with an emphasis on ecological restoration process. Twice he has won the Young Scientist Award, one from the Government of Kerala in 2000 and from the Indian Association for Angiosperm Taxonomy in 2002. He joined us in 2010. He is currently the principal scientist of the Tree Physiology Department, and he is also the program coordinator of Sustainable Forest Management Division. A scientist with nearly 60 research publications, he has authored books on endemic trees and medicinal plants. His current research interest includes climate change impact studies, seed physiology, restoration of threatened trees, and non-timber forest products of the Western Ghats. With immense happiness, I welcome Dr. Jose to this particular function. Yesterday we had two talks. One observation is that there was absolutely no questions asked from the audience. I think today will be a different day. There will be better interactions. Dr. Jose is going to speak to you about a very interesting topic, the science of saving threatened trees in the Western Ghats. As you might know, all the plants in the Western Ghats are not equally abundant. There are some trees which go into the rare category, then to the threatened category, and then finally to the extinct category. There is a species extinction happening. Now, Dr. Jose, for a many number of years, Dr. Jose was trying to save them, to save plants from trees from extinction. How do you do that? What are the information you need to protect or save a tree? And how do you actually do it? And he will also be talking to us about the success stories of saving trees with his research. So, Dr. Jose.
Very good morning, all of you, and welcome to my presentation. It's titled as The Science of Saving Endangered Plants of Western Ghats. The presentation consists background information about the species diversity, endemism, concept of rarity, IUCN status, criterion and categories, further identifying the causes of rarity, and the information or knowledge required how to save these species, and the work approach, the study methodology to reach the goal, and finally the case studies that we have conducted through different projects and success stories. As you know, Western Ghats, India is one of the 36 hotspots and one of the 8 hottest of hotspots of biodiversity in the world. These figures indicate that the immense biodiversity in terms of plant, fauna, microbes available in the Western Ghats region. It's running parallel to the west coast of peninsular India, starting from the Tapti River in Gujarat, flowing down through Gujarat, Maharashtra, Goa, Karnataka, Kerala, and Tamil Nadu through a distance of 1,600 kilometers. The area, the green landscape, have high featured in topography, particularly the altitudinal variations, normally from 1,000 meter to the highest peak 2,695 meter in Animadi Shoalai, and rainfall from 2,000 to 7,450 mm per year, and temperature and varied soil types from laterite, red loam, medium black to red gravel. And these topographical features, the amount of rainfall, the altitudinal variations, and the soil types resulted different vegetation formation in the Western Ghats, from tropical evergreen, semi-evergreen, moist deciduous, dry deciduous, shrub jungles, mountain grasslands. Apart from these vegetation types, Western Ghats also hosts some edaphic vegetation forms like Myristica swamps, mangroves, bamboo and reeds, etc. Finally, it contributes around 30% of flowering plants, that is around 7,500 species of flowering plants of the Indian subcontinent, of which around 2,300 species are endemics. Coming to the part of Western Ghats, Kerala, the area, as you know, the area is more than 38,664 kilometers square. However, it holds 1.18% of the Indian subcontinent, but it supports more than 29% of the flora of the country. The flowering plants around 5,200 species, of which 4,300 species are indigenously staged. Around 1,600 plants which are endemics to the peninsular India, and around 900 plants are endemic to the Western Ghats. Among the endemics, around 270 species are exclusively seen in this Kerala state, and of which 53 species are woody plants. Regarding the threatened species composition, around 550 plants are reported from the Kerala part of Western Ghats, of which nearly 260 species are identified or recorded by the IUCN for different threat categories. Among these exclusive endemics, 100 red listed species, IUCN red listed species are reported from the Kerala. As you know, this study is species oriented. So species is the fundamental unit of an ecosystem. It's lost due to various factors like physical and biological reasons, or exploitation, habitat loss, biological constraints, makes severe impact on the species integrity, composition, and functions. Interrelationships between each organisms and alter ecological functions and lead degradation process. So in this particular ecosystem or the various landscapes, trees, non-timber forest produce plants, medicinal plants, and reptiles are become the most vulnerable groups because these species are highly exploited for their various economic attributes. So in this background, we can say, what is the rare species? According to Drury 1974, he defined as the rare species is one that occurs in widely separated small subpopulations, so that interbreeding between these subpopulations seriously reduced or restricted to single populations. So what happened is, in the Western Ghats region, the human degradation, human interventions made fragmentation of landscapes. So the populations become fragmented into become smaller and smaller groups, so that individuals becoming later leading to the rare category. And once the species become rare, the extinction process begins. So extinction is not an abnormal fate in the life cycle of a species, but once the environmental degradation increases beyond its level, the species could not get evolutionary time span for its normal evolutionary process, and that leads to the untimely extinction in a species. According to IUCN, the current species extinction rate is between 1,000 and 10,000 times higher than it naturally occurs. So, as per Rabinovich in 1986, he given 7 forms of rarity identified. So that is based on the species with the larger geographic range and species with a small geographic range. While we seen these different concepts, we can compare the species in our Western Ghats, and we can see that once the species narrow habitat specificity, locally abundant in specific habitats. So in the case of, I am pointing out some of the examples belong to the Myristicaceae family. Here we can see the Gymnacranthera, Myristica bedomi, or Myristica malabarica, whatever it is in the Myristicaceae. This is a typical example. These habitats are specific. They are seen either in the Myristica swamps or in the evergreen forest areas. but it is locally abundant in that habitats. So that kind of equation is designating a particular species to a rare species. Another definition is narrow habitat, but constantly sparse in specific habitat. For example, in the case of Garcinia morella, we can take an example. It's a narrow habitat specificity, but it is sparse in their habitats. Number that can be very less. Similarly, different concepts have given so that we can equate with our many of the species and found that this species deserves this rare status or the different conservation priorities. For example, in the case of one of the significant species, Coccineum fenestrata, you know, the Maramanjal. This is a wide habitat specificity from India to Bangladesh and Sri Lanka, but this constantly sparse in several habitats. In its habitat, the species are become sparse or lesser because of the over exploitation. Similarly, species with a small geographic range, we can see some species, even it is included in the rare category, have wide habitat specificity. For example, Hopia parviflora, it can seem distributed India, Sri Lanka, and beyond that, but it is locally abundant in several habitats. Even though its extension is higher, the species have the area of occupancy and other factors considering the species are included in the rare category. That's the definition gives this particular species. Some species have wide habitat specificity, but constantly sparse, and some are narrow habitat specific, locally abundant. For example, Ochrinochleum is a riparian tree of the Western Ghats. It's a narrow habitat specificity. It's typically originally seen in the riparian areas, but it is abundant in certain these habitats. So that species can also be considered under this rare category as per Rabinowitz. So in short, what are extinction prone species? Small individuals populations are subject to large fluctuations. Once this fragmentation is taking place, species become isolated and become settled in their niche. So the species the adaptability of the species will be decreased. The tolerance level, the different environmental responses, the tolerance level of the species will be reduced. And another feature is that the low frequency of flowering. The species will show low frequency or irregular flowering period or long intervals in flowering. Such species are also indicating that it going in the extinction process. Similarly, the seeds with the low viability. As you know, the recalcitrant seeds with the low viability period, it's also an indication that species have the soil seed bank as well as the seedling bank are in the non-viable state, and it is moving in the extinction process. And some species have very poor dispersal mechanism. The seeds will be sentient and following the floor itself. There is no specific mechanism. So such species will also be in the moving strategy of this extinction process. And some other are poor colonizing ability. There may not be a vegetative expansion of the species. There may not be a viable seedling regeneration. So that kind of species are also coming under the moving in the extinction process. Based on these various things, IUCN and the international agency were responsible for assessing the conservation status of this flora and fauna, and different threat status for this species, of which 7 threat categories were defined. Among these, critically endangered, and vulnerable categories are the most threatened categories for species conservation. So within these, species are also moving to or already moved to extinct. It is no longer to exist. The species is totally wiped out from the earth, and the species is categorized under the extinct. And some are in the extinct in the wild. That is, natural populations are depleted, but the species is available in the cultivation area. For example, the Camphora galanga or the Saraka shoga, the species or natural populations have been wiped out. But in the case of critically endangered species or endangered or vulnerable, species are very much danger of extinction. But in the case of critically endangered, it needs immediate future extinction will be taking place. For example, I just pointed out 2 species, that Anacorsa densiflora Syzygium palacotton. These species have no study records, and there is very little information about the species, and the species to be considered in an immediate future. That is the point we have to stress on, that is critical endangered species extinction will take place in the immediate future unless studies have been initiated. And in the case of endangered species, species will move to high risk of extinction in near future. And in the case of vulnerable, the species will move in the medium term future. This is the rating of the species about their extinction and the untimely endangerment of the species. So based on this IUCN, some criterion were followed how to place these threat categories. And as per a critically endangered placing, the species, the extent of occurrence will be less than 100 kilometers square, and the area of occupancy will be less than 10 kilometers square, and the number of adult individuals will be 250, less than 250. And beyond that, there's some species degradation rate in the last 10 years, if it is more than 80%, such a species, such distributional features or species can be considered as a critical endangered. Similarly, the endangered species, the extent of occurrence will be higher than the critical endangered, area of occupancy will be higher, number of mature individuals will be higher, rate of decline will be less. So this way the species are categorized, and we are prioritizing these critical endangered, and vulnerable species for various kinds of studies. So this is the background of the species recovery research. And now we are coming to how these plants become threatened. What are the major reasons for untimely rarity of the species? We can see that there are 5 major broad areas of the causes of rarity. One is the physical induced or human induced factors, ecological factors, biological constraints, genetic parameters, and climate change impact. These are identified as the major causes for the untimely endangerment of the species. Coming to the physically oriented or human induced factors, at least a dozen factors are influencing or originating from the human side. That includes habitat loss or modifications that I mentioned, that small individual populations created, so that will create an island effect of the species. You know, once the species become in that island, the tolerance and the adaptability of the species will be less. So that there is known as island biogeography. So many of the tree ferns, rhododendrons, magnolias, orchids are now in the stages of habitat loss or modifications. And developmental activities like construction of roads or dam construction, and mining activities, particularly sand mining in the riparian courses in the southern Western Ghats, led depletion of many species. As you can see that this is the habitat of Ocrinochloe missionis. It's a typical riparian species. The sand mining in these habitats created the depletion of or removal of trees from this species, and gradually the slight rise in water in the northeast monsoon period or in the southwest monsoon, the trees will be easily removed from these habitats. So sand mining is one of the important reason for originated from human on depletion of many of the riparian tree species, overgrazing, and grass reclamation programs for the plantations, and overcollection of species because of the various economic and medicinal attributes. Many species are exploited. And in addition, the pressure from the exotic weeds. Many exotic weeds are spread in the natural habitat of the species and suppress the growing seedlings, and thereby many of the seedlings, the regeneration of these species is getting arrested. So these are the major factors originated from the human. And another barrier is on the ecologically related ones. That is, many factors are involved in the ecology of the species. One is on the low population size. Number of adult individuals very less in that cases. So the number of adult individuals is one of the major factor assessing or prioritizing the species conservation, and the low number of younger individuals. You know, in a population, there are different age classes are there. Mainly the reproductive classes are there, pre-reproductive classes, and post-reproductive classes are. pre-reproductive classes or the younger trees are the indications of the populations is growing or increasing. So in a population, the number of pre-reproductive individuals are in lesser in number. So the population is somewhat facing poor vigor and retarding the growth. And a low abundance in a landscape, distribution range species will be analyzed with the population studies. So the abundance values, the abundance in terms of the frequency, density, and dominance, the IVA, important value index, will be calculated. And that value is showing that poor values of many of these threatened species. So the low occurrence of the species indicating the low abundance. And that is one of the another reason, ecology related reason on the reduction of these populations or the species in their habitats. Another thing is on the poor soil seed and seedling bank. That is, some species have very poor dispersal mechanism and some have very poor fruit production. So automatically there is a poor soil seed bank is there. Some may be dormant for a long time. Some may be germinated, but the seedling may not be survive in a better position. So this kind of poor soil seed bank, seedling bank are also related to the ecological constraints of the species. And I already mentioned about the low ecological amplitude. The adaptability of the species will be less. The tolerance level will be less. Such species, such feature also leading the ecological reasons for the threat of the species. Another is on the distinction of the distribution of the species. Similar species may be seen in dissimilar geographical situations. For example, in the case of Gutta, there's a major stock is distributed in the Malayan region, Indo-Malayan region. But species have been dispersed or distributed to different continents due to the continental drift theory, according to the theory. So this is the distinction distribution of the species that leading to the endemism of the species. So in that cases also many species facing this ecological constraints and their survival, and they are very much susceptible to the climate change issues also. So these factors are leading ecological constraints of the species related to the threat of the species. Another major constraint is on the biological factors, the reproductive biology of the species to be analyzed. The low frequency of flowering. In the case of dipterocarps or many of the legume trees, can see the irregularity in flowering. So the irregularity or long intervals, sometimes 2, 3, or 5 to 6 years will take for a peak flowering. So this kind of indications is leading to the availability of seedling collection. So everything will be disrupted, arrested. So that is one of the major biological constraints of the species leading to the rarity. Another is on the seeds with the low viability. For example, many of our evergreen species and riparian species are in the categories under the recalcitrant category. Recalcitrant seeds are very short viability period. Its viability will be lost within weeks or days. So the climate change related issues will lead to the major bottleneck for the viability loss. And such species, such recalcitrant categorized seeds bearing species are one of the major constraints for the survival, and that lead to the various threat stages. Another is the low dispersal ability, leads chances of self-breeding. Once the species occupied in isolated niches, the individuals will cross-pollinate, the individual will self-pollinate, because there may not be enough space for, or may not be different individuals in different locations. So the similar individuals will try to breed. So self-pollination will take place. So in such cases, the seedlings will show some genetic characters like albino formations, or some kind of adaptations will take place. So that is also because of the low dispersal ability of the species leading to self-breeding. So that are also leading to the extinction process. High degree of fruit and seed predation. Many of the species have the fruits are highly predated by various mammals, birds. And so this leads to the arrest of the soil seed bank, and that may arrest the regeneration soil seed bank. So for in the case of various diasporous species, Disosalum, the hornbill are the major predator. Atuna indica, these are highly predated by various birds and mammals. So that also leading to the biological threat of the species. And in some cases, absence of pollinators for effective pollination, that will be arrested. And the colonizing ability, sometimes the species may not have a vegetative sprouting and the seed may not be in a viable stage. So these things will lead to the threat categories, and these are all categorized under the biological threat of the species. Within this biological threat, various pest and diseases are also happen in the many species. This is the natural mechanism. However, on studying various species, we can reveal that. In some examples I quoted here, in the case of Gluta travangurica, the Indian redwood, and it's distributed in the southern Western Ghats, Kerala and Tamil Nadu. In Kerala, then a sanctuary is named after the species, that is Shendurani Wildlife Sanctuary. The seeds are infested by mango nut weevil. So this lead to the loss of many viable seeds. And in the case of this Humboldt-shea wileyana, there is seed borer is there. And in the case of Ochreunicolium missionis, we can see these fleshy fruits often associated with these fungal pathogens, and they degrade the pulp along with the minute seeds. And in the case of this Disocelum malabaricum, the both aerial and ground level predation is there. And Calophyllum, premature abscission. And in Gluta, the inflorescences are highly predated, or the flower is damaged by hopper incidents. And Mimicillon lavzoni, that is, seeds are highly infested. So various pests and diseases are also happening on the life cycle of these species that lead to the poor seed regeneration, seedling regeneration, and leading to the threat stages, threat categories. So regarding the genetic factors, while we analyzing the genetic factors, the low population size that resulted due to various reasons like environmental variations, catastrophic habitat destruction, several human-induced reasons lead to the fragmentation of the species. So the lower effective population results more demographic variations. The population size will be restricted. The number of seedlings will be reduced, the adults will be reduced, and this further lead to the different genetic issues like inbreeding depression. The number of different cross-pollinating possibilities will be arrested. Species will breed themselves. So that will lead to the inbreeding depression. Inbreeding depression will lead to the genetic drift, and finally to the poor genetic vigor of the species. So this will lead to the extinction of the species. So these are the factors related to the biological constraints and genetic factors leading to this species threat process. And finally, the recent studies indicating that climate change is also affecting this or leading to the threat of many of the species. In the Western Ghats, we had a recent study on this species with respect to the climate change while analyzing the 25 years back data. Many of these species facing or showed various changes in their phenofaces, reproductive biology, and biotic interactions. So these factors are considered as manifestations of evaluating the climate change on forest species. This change in climatic factors results the phenology calendar of the species. You can see a phenology calendar of Gluta travangurica depicted here, where we have noted drastic changes in the phenophases. Species exhibited a delayed flowering because the species normally needs a warm humid condition for their flowering during January months, but the erratic or absence of summer showers lead the delay in flowering. So delay in flowering lead to the delay in fruit initiation, delay in fruit senescence. But in many cases, the species, once susceptible to climate change, also try to manage this climate change impact by adapting some resilient strategies like extended flowering or Sometimes this will manage with the monsoon season. The fruit will be dehydrated during onset of monsoon. So once climate change is there, it affects the phenology of the species. It affects the reproductive biology. It affects the biotic interactions, especially the pollinators, dispersal agents. But the species will try to manage this to an extent in the form of adaptations. So once the pollination, once this species normally flowers in just during summer season, March, April, but the climate increased temperature maybe started flowering a little bit earlier. So the pollinators depends on the species, the rhythm of the pollination or the synchrony of the population will be affected. So similarly, the fruits, once the mammals or the biotic agents attach to the species, while visiting the species, the fruit may not be ripened. So that occasions these agents will be moved to some other species. So the entire life cycle of the species will be changing. So that is affecting the natural regeneration, effective pollination process. And in the case of dioecious species, the synchrony of the flowering will be upset. Normally the male and female flowers starts flowering simultaneously, or little bit male maybe a little bit earlier, but it will be long, it will have a long flowering time. But once the climate affecting the synchrony of the flowering time affected, so may not cope up with the pollination process. Either male will be in advance or female will be delayed. So at a similar time, both flowering will not be happened. And in some cases, the anthesis time will be changed. We have some observations on some other species in the Persia and Crinoclemisionis glutatraumagorica. The anthesis time has been changed from night to daytime. So what happens is the stigmatic receptivity then changing. The pollinators visibility will be changing. Some cases, there are some advantages are also there. While we change, while this anthesis time changing to daytime, more insect visitors are there. Butterflies are coming. So that way the species is adapting these constraints. And the fruiting and seed episodes are also varying. So several studies are in the launching stage, indicating that climate change is also making serious changes on the natural dynamics of the species and subsequent rarity of the species in their ecosystems. So while we moving to what are the information we require to save this species? That is the second stage of the study. What kind of information we required for rescuing this species? First of all, we should know the distribution range of the species. Then only we can identify the populations. We can assess the population size. Everything can be generated only after a distribution study. Then there is a clear-cut ecologic studies are required. What is the population structure of the species? How many adult individuals are there? What is the seedling classes? And what is the relative dominance of the species among its associates? And next is on what are the in situ requirements required for the species? What are the microclimate required? What are the soil it prefers? And what are the biotic agents associated with the species dynamics? What is the natural performance of the species, natural regeneration performance of the species, whether the species will clonal regeneration ability, what is the seed biology of the species, what is the seed type, and what is the normal period of viability, how long it will be viable in the ambient conditions or in the extended or in the controlled conditions, and what is the critical moisture content. So this kind of information is required for its germplasm storage of the species. And what are the pests and diseases associated with this? So once in the vegetative phase, which are the insects associated with feeding these leaves, and which are the insects are visiting these during flowering time, and what are the other species related to sometimes there may be fruit predation is there. So this kind of documentation of each and every biotic interactions and related to pest and diseases, fungal infestations, Sometimes the fruits may be fungal interaction may be there. Sometimes the seed may be infested. So this kind of documentation is required. Then only we can go for the rescue study. And thereafter, we need to restore the species. For any kind of restoration programs, we need a suitable planting areas of this species. For this, niche modeling of the species is required. So niche modeling will enable to identify suitable habitats, probable suitable habitat of the species. After finding out the suitable habitat of the species, we have to find out the microsite conditions required for the restoration. Even though in a landscape which is suitable for the species, microsite conditions are very specific for the survival of the seedlings. For example, while we are planting seedlings, the canopy gaps, the moisture soil, and even the conspecific association, these things to be considered. Then these are all found to be affecting the survival of the seedlings. So this kind of information is required for any recovery program. So we have developed a methodology for this. This have been prepared in a flowchart, that is a population survey along with the niche modeling. And this needs literature study. And we also recorded the area of occupancy, extent of occurrence, number of adult individuals. The population ecology, there is a population structure, that is how the species are distributed in the forest ecosystem, how it is spatially distributed, in which strata it occupies, what is the frequency of the species in the respective ecosystems, what is the IVA values, what is the age class, how percentage crowns are spreading, and what is the population dynamics, that is what is the leaf phenology, what is the duration of the leaf development, maturity, senescence in such incidents. So this kind of information that we already mentioned or discussed are to be covered for achieving our goal. So apart from this, after identifying the reasons of rarity of the species, we have to develop appropriate conservation protocol of the species. That includes the propagule collection from source populations. Source population, you know, that is the species with the allied trees having these pest and diseases will be less, will be more peak fruiting, or viable seeds will be there. So propagules will be collected from such populations, and the seed biology and vegetative propagation, nursery technology, plant stock production will be carried out. And finally, the species will be transplanted to their suitable areas identified through niche modeling, and the seedlings were planted. So that process, the restoration process, involves both reintroduction and restocking. In the reintroduction, the species, in the past, the species was there, but due to the various changes or habitat loss, the species have been wiped out. So such areas where the seedlings will be planted is known as reintroduction. And restocking is the degraded populations where we are enriching the individuals to a safe level. That is, we are known as restocking. The seedlings were planted and we will monitor the seedling survival. There are many factors may affect the seedling survival. So that kind of information is very specific to species. And finally, the species, this information will be submitted for a reassessment of the conservation status of the species. That is the ultimate target of the entire species recovery program. So here I carried out a demonstration how this data is collecting. So first task is on the distribution ecological studies of the species. First of all, the population survey of the species will be carried out in its distribution range. That is based on the literature, herbarium, and field visits. And the populations will be mapped out. The extent of occurrence and area of occupancy, mature individuals, seedling class, habitat threats, everything will be documented. Based on this, in the case of the Saporosa bodloni, 3 populations are reported. And this way, this distribution study will be completing. And thereafter, this niche modeling based on the Maxent software is doing. So based on the 19 climatic variables, the suitable habitats of the species identified in the Western Ghats, the red color indicating the suitable habitat of the species. And thereafter, the ecology of the species that is included the quantitative analysis of vegetation data and abundance studies. Within the quantitative analysis of vegetation data, that is find out the stratification, horizontal distribution, age-wise distribution, crown projections, and in the abundance, the GBH height relationships and frequency, density, dominance, etc. will be worked out. So finally, after this structural analysis, profile diagrams will be prepared based on to identify the stratification of the species and the number of age classes, adult individuals, pre-reproductive individuals, such as informations will be collected, how the species is distributed in the ground, spatially distributed, whether it is in a clump pattern or uniform pattern or in an irregular way. So whether it is in slopey areas, how the seeds are on senescence moving. So this kind of representations will be collected during the ecology study. And this is a data sheet of the population structure data collecting, that is the GBH, basal area, age phase, and populations are permanently marked for monitoring. And finally, the important value index of the species were collected among the associated species. For this, quadrates were identified, quadrate size were identified as per species area curve methodology. Normally 15 to 20 meter square plots are taken, and within these plots, individuals above 30 centimeter will be counted, and each species were worked out about their frequency, density, dominance, and finally the IVA values were calculated. And regarding the population dynamics, that is the natural dynamics, both the vegetative and reproductive phenology phases are covered. You can see the vegetative flushing of the species, in which the caterpillars may be infested. So the adult will be identified. So that will be records of the species host relationships. In the reproductive phenology, from flower bud to regeneration episodes will be worked out. And another important aspect of the reproductive biology is the floral biological studies. Here I shown one species that is in the Ocrinoculum missonis, how the floral biology studies are doing. So you can see the number of flowers in a particular. So this is a flower head in a flower head. You can see the normal flower buds with the protruding stigma. And this is the corolla lobes, and this is the pistil, and this is the LS of flower. Here, normally the anthesis is taking place during evening hours. So from 6 p.m. onwards, the stigma is gradually moving up. And after half an hour, it reaches the central position of the corolla, and gradually it touches the corolla throats, where anther lobes are situated. So afterwards, the anther lobes dehisce with a thick load of pollen grains. It gradually reaches out. So around from 6 to 8 p.m., the corolla is fully opened with a thick load of pollen grains. The stigma protrudes outwards. So at this time, the stigma is melanoreceptive. So it inhibits the self-pollination of the species and permits for cross-pollination. Thereafter, stigma will be viable so that to facilitate the cross-pollination of the species. So this way, the floral biology of the species to be worked out to find out the breeding behavior of the species. So this is one major aspect in the reproductive biology study. In the in situ requirements for the climate and soil studies, the atmospheric temperature day and night, the canopy temperature, humidity, rainfall are also to be recorded. And the edapology, texture, soil texture, moisture, soil temperature, pH, nutrients are also will be analyzed. And this is the data sheet of this recording of this climate and soil. And then we are coming to the conservation strategy, developing the nursery technology of the species. What is the plus tree selection and the collection of propagules, auxin treatment, and these ramets are produced through different vegetative propagation methods. And the low-cost nursery conditions are utilized for this propagation of the species, where high humidity and low temperature are found to be favorable for seed germination and the rooting process. And seedling morphology are also worked out. Some abnormal seedlings may be there. So that can be treated in the form of albinos. So these are all indications of the species leading to the rarity. In the case of some species, in the case of Glutatravangorica, the major observation is on the viviparous condition. Normally, vivipar is reported in the mangroves, but this evergreen species shows an indication of vivipary, so that we analyzed it. So it is an indication of this climate change on the species, because normally the seeds are falling during January, February months, where the shola patches having this water availability. So during climate change, the sholas are dried up. So seeds could not germinate as it is recalcitrant nature. So the inborn character, like many of these species related species situated growing in the swampy areas in Indo-Malaya, the species trying to agentic adaptation is exhibited in the form of vivipary. So that is the indication of the climate change studies on the Glutatravangorica, the viviparous formation. And this is the data sheet of the vegetative propagation experiment carried out in different rare species. And this is the ring layering success achieved as part of duplication of the species. And this is the seed study carried out. In a normal seed biology studies, you have to find out the initial moisture content first. That is, the moisture content is the critical role for germination of the seeds. And thereafter, the seeds were categorized as per the moisture content. The critical moisture content will be estimated. And normal viability period. This is a case study carried out in the wild nutmegs of the Western Ghats. And what is the days required for initial germination of the seeds? What is the type of germination? While after this categorizing the seeds are stored in different climatic conditions in a seed bank. Like if it is recalcitrant species, 20 degree, 40 degree, 40% RH. And similarly, different climatic conditions are set in a seed bank. So the seeds are kept in different storage conditions using different containers like polythene bags or polycarbonate bottles. And periodic viability test will be carried out. And the most suitable conditions will be standardized for its long-term storage of the seeds. Normally, the recalcitrant seeds will be viable up to 6 to 1 year maximum, but orthodox seeds will be long viable. So for this seeds to be reduced EMC, you have to reduce the EMC and store the seeds in low temperature conditions, freezing temperature conditions. So that way we can extend the viability period of the seeds. And I also mentioned about the seedling morphology of the species. Many of the species having abnormal seedlings, sometimes maybe albinos. Albinos, I told, that is an indication of a genetic marker of self-breeding. And sometimes there may be twin seedlings or multiple seedlings are there. So you have some genetic importance, breeding scope is there. Some haploid, diploid levels are there for this multiple embryos. And finally, the propagation study leads to the plant stock production. And thereafter, the seedlings will be transplanted to appropriate areas after identifying the suitable areas through finish modeling. And the seedlings are planted with the display boards are installed, demarcating the planting area. And the seedlings will gradually monitored for a long time, at least for 5 to 6 years. So here we can see this Dipracarpus borlonii, a critically endangered species growing in different forest patches after it is now attained 10 to 12 years after planting, and Humboldt borlonii, another critical species. So this is the methodology in attending the species recovery program. It's a multidisciplinary study, needs expertise and needs a lot of lab manpower is required, a lot of expertise is required. So such a way we can conserve or recover a species from its untimely endangerment. So we have conducted several projects on this line. I can show you some of the results on this. This is one of the Glutatravangorica and Ocriniculum missionis. It's part of my PhD during 2000. And this is another DBT funded project we have carried out on the Dipracarpus borlonii and Humboldt borlonii. So two critical endangered species. This Diplocarpus boloni having high embryo problems, that viable embryos are very below 10%. At the same time, this species having various other problems also. So it's a very important species. And another is on the Drypetes, Hypnocarpus, and many other medicinal trees like Empelia, Canarium, Cosinium, Garcinia morella. These are all investigated under this line of research. And wild nutmegs like Malabarica, Bedome. Fatwa is a very important species seen in the Kultupura Valley. It's a marshy species. And we have also conducted some studies on the tree legumes, that is Cyanometra bedome and Chytodendron pinnatum. Chytodendron pinnatum, where in the past, this tree wood, from the wood, the oil is extracted, that is known as kulavanna. It's also known as senna pine. And Aporosa bodloni, Drypetes confertiflora, Inga cyanometroids. These are all carried out and studied through different projects. Another project on this the species recovery, including the genetics in Actuna and Hypnocarpus longipedulatus. This is an ongoing PhD program. And this is the case studies we have carried out on different species. And afterwards, we also prepared data for reassessment of the conservation status. So around 17 species, we have submitted data for IUCN for status reassessment. That's about the species recovery program. One thing I want to tell you that the last flooded conditions during 2019 and 20, the riparian tree Ochronychum missonis are seriously damaged. So this kind of information is very useful. So we had to take immediate studies, field studies on management of the species by planting seedlings in the riparian course, at least in the protected areas even, that is also possible. So that's about the species recovery program. And I am happy to receive your queries, your doubts, anything you can ask now, so I can answer to you. It's an important area of study.